How's it going, everyone? It's Chow here, and today we're going to talk a little bit about the cell cycle, so let's get started. Alright, so the cell cycle is generally going to be broken down into sort of 3 sections, 4 sections, maybe 5 sections, depending on which type of chart you're looking at and which figure from whatever textbook you're looking at. But this is generally one that you're going to see pretty commonly. So you have the G1 phase, the S phase, the G2 phase, as well as your actual cellular division phase over here, which includes mitosis, as well as the G0 phase over here, where the cell cycle is actually at rest. So this is the general cell cycle. When we're talking about a cellular cycle or cell division, this is what we're talking about. Again, keep in mind that cell division itself is really only happening in mitosis. So that's cool and all, but when you look at the cell cycle, obviously we have to talk about the requirements that you have to go through and need in order to push that cell into the cell cycle. So oftentimes what you need is this over here, which is a reproductive signal, which is just basically a signal to initiate the cell to basically divide, to go through the steps, to go through division, right? You obviously need DNA replication. Remember, if you're going through mitosis, right, you need to make 2 cells from one original cell. And as a result, the 2 daughter cells each have to have the same amount of DNA. And in order to do that and maintain that, you need that original genome to be duplicated. Obviously, you need segregation. So after you copy that DNA, you need to separate that DNA ultimately into 2 new cells through segregation. And then, of course, cytokinesis is where you're actually physically splitting the cell into 2 new cells. So here are just some brief requirements that you need for a cell to divide. Pretty straightforward, I think. Of course, it gets a little bit more complicated because we have different types of cells. Prokaryotes, which lack membrane-bound organelles and lack a nucleus, generally require external factors, for instance, a nutrient concentration or environmental conditions, to basically push them into the cell cycle. So in the presence of a lot of food supplies or nutrient supplies, the cell's division cycle is often going to be sped up. However, what's fascinating about prokaryotes is that because they usually only have a single circular chromosome, for instance, if you think to bacteria, they have that circular strand of DNA. It means that it contains that origin of replication and other sequences. And because it's just one single circular strand, it's much easier to divide. So at the end of the DNA replication, the daughter DNA molecules are just going to be pushed towards the end of the cell, and the cell just kind of splits, goes through cytokinesis, mitosis and cytokinesis, and that makes 2 new cells. So in terms of prokaryotes, the whole cell cycle is a little bit more straightforward. They aren't limited by membrane-bound organelles, and that means that prokaryotes tend to replicate fairly rapidly, which makes sense because if you have an agar plate of E. coli or some other bacteria, it tends to replicate fairly quickly compared to, say, mammalian eukaryotic cells. So that should be pretty straightforward. And if we look at eukaryotes often require some kind of a reproductive signal for cell division, and these signals are going to be related to the needs of the organism. So what that means is that sometimes you have cells within the body that really does not divide at all, and there are examples of such present. In addition, the signals themselves often depend on the needs of, for instance, development, for differentiation, like in the case of stem cells, in the case of defense, maybe in your B cells, for instance, or in deterioration. So if there's deterioration, you want to replicate to replenish whatever tissue, for instance, that's actually deteriorating. And as a result, along with all these factors over here, and the fact that eukaryotes have membrane-bound organelles, it generally takes longer for them to divide. And also the one thing that makes it maybe even longer and longer for them to divide is the fact that they have multiple chromosomes in many cases, and their chromosomal DNA strands are also going to be larger. So it's going to take longer in most cases for eukaryotes to divide compared to prokaryotes. So that should make pretty straightforward sense. But if we want to go one step further, we want to look at the cell cycle itself. So the cell cycle is just simply the period from one cell division to the next. In terms of the cell cycle, you have sort of the main division, right? That's broken down, and that's the mitosis section as well as the interphase section, which is shown in green over here. And then most cells that are found within your body, for instance, are not in mitosis. So they're not going to be in that active state of dividing. They're actually more likely to be in the interphase, which involves most likely growth or maybe replication of DNA. So there's that. Now, if we move on and look at the individual phases themselves, one key thing you'll notice is that interphase is broken up into a couple of phases. You see the G1 phase or the gap 1 phase. You also see a G0 phase, which is actually not shown here, but is actually shown in this particular figure over here. So you have a G0 cell cycle arrest phase, along with an S phase, which is where actually DNA is being replicated, and then a G2 phase, which is just a little bit of machinery assemblage for mitosis as well as further growth. So there you go. Interphase is being broken down into sort of multiple subphases. So let's take a look at the gap 1 phase just very briefly. G1 phase occurs between cytokinesis and the S phase. So cytokinesis is over here and the S phase is over here. This might be a better chart or diagram to look at. So you have cytokinesis, S phase, G1 right in between. Now, this is a period of growth for the cell. So the cell is just growing, not quite ready to divide. It's just growing, maybe taking up nutrients, making machinery if need be. And the duration is quite variable within the G1 phase. It can be a few minutes to sometimes years. And some cells will actually not go through the entirety of G1 phase and into S phase, but actually rest and stick around in this phase, which we label as G0, also known as the cell cycle arrest phase. So what's really key perhaps about the G1 phase is that towards the end there's a restriction point, also known as a cell cycle checkpoint. And basically it's over here somewhere, and if the cell decides to go through that restriction point, right? So it says that it's ready to divide, it's ready, it has all the machinery for now, and it's going to jump over that restriction point at the end of G1 into the S phase, it means it's going to be committed to DNA replication. So if the cell decides to go through the restriction point at the end of G1, it's committing itself to replication. Cool? Okay. Now, once we enter into the S phase, so remember, it's committed, it's going through that checkpoint at the end of G1, and it's entering the S phase, we now have to replicate that DNA. Remember, through mitosis, you're going to make 2 cells. You want the same amount of DNA in those 2 cells. So in order to do that, you need to replicate that DNA, which is what happens in the S phase. So the number of DNA is doubled, but the number of chromosomes remain the same, because you have 1 chromosome here, 1 chromosome here, 1 chromosome here, and 1 chromosome here. So the chromosomes that are present are basically labeled in different colors. You have one sister chromatid over here and another sister chromatid over here, and same goes for here. So this is a sister chromatid, this is a sister chromatid, but this entire thing, this purplish blob X over here is a chromosome, and this blue X is another chromosome, and this is also chromosome, and this is also another chromosome. So this is where the terminology might get a little bit tricky, but in S phase, you're basically kind of doubling the amount of DNA present in that one cell, but you're not doubling the number of chromosomes, if that makes sense. So there you go. So you have right now 1, 2 chromosomes here and 1, 2 chromosomes over here. So S phase, replicating that DNA. Now, once you go through the S phase, you get to G2, or the gap 2 phase. And this is the phase where the cell basically prepares itself for mitosis. And it's nothing more than another period of growth and also production of cellular machinery that's necessary for the cell to divide. So once again, What's really fascinating is that you have these checkpoints also that the cell needs to get through in order to reach the actual division phase, which is mitosis. Side note is the G0 phase, and this is the phase where the cells are no longer actively dividing, so it's not basically trying to push itself beyond that G1 checkpoint into S phase, but it's just kind of staying in this one phase over here where it's not really trying to commit itself to cellular division, and this cell cycle arrest phase is also known as the G0 phase. Cool. So restriction points, or Rs as we sometimes call them, restriction points basically are present in the cell, and it's generally present before the G1 to S phase transitions, right? So we talk about it over here. You have this restriction that's present, and what this basically allows the cell to do is basically check up on itself to make sure it has everything it needs to move on to the following phase. And this restriction point at the G1 and S phase, so in the boundary between the G1 and S phase, is basically that step again that's going to push the cell to commit itself into replication. And specific signals actually sort of trigger that transition from G1 to S, as well as other transitions as well, for instance, from S to G2. And it's regulated by this family of cyclin-dependent kinases, or CDKs, along with those cyclin complexes that kind of work through allosteric regulation. So allosteric regulation is binding, right? And through that allosteric binding, you can regulate the pushing on of that cell cycle or the stopping of that cell cycle. So in the basic gist, you have a CDK that forms a complex with cyclin, which ultimately phosphorylate a target protein. And then, of course, different numbers of CDK families exist at different phases and checkpoints. And you can probably read your textbook and read up all about CDKs and cyclin complexes, but I'm not going to get into too much detail about them right here. So there you go, restriction points. Very critical to make sure your cell is ready to move on to the next phase. Now, in terms of cell cycle checkpoints themselves, we obviously talked about the G1 checkpoint, but there are really sort of 4 different cellular checkpoints that are triggered by different conditions, and it's also meant to basically prevent the cell from entering the next stage of the cell cycle if it isn't really ready for it. So we have the G1 checkpoint, which we discussed, which is really sort of just a DNA damage checkpoint to make sure the DNA which we are about to replicate is not damaged. Very important, obviously. The S checkpoint is basically checking for incomplete replication or DNA damage. So once you replicated the DNA, you want to check to make sure that it's replicated properly and there aren't any issues. The G2 checkpoint, again, is going to be looking for DNA damage. So before you actually actively go through mitosis and cytokinesis to separate that one cell into 2 cells, it's checking to make sure that there aren't really any DNA damage. And finally, you have the M checkpoint. So during the M phase, there's another checkpoint that just kind of checks to make sure that chromosomes are basically attached to the spindle. And so if the chromosome's not attached to the spindle, then the cell cycle is going to stop to make sure that the chromosomes do end up actually being attached to the spindle and that everything is working properly. And so ultimately one cell doesn't accidentally get like more than one copy of the necessary chromosomes, or the other cell would get like one particular chromosome less. So just checking for whether chromosomes are attached to the spindle or not. So you ultimately want the chromosomes to be attached to the spindle. So here is a great diagram, right, to make sure that the cells are being pushed on. So again, G1, there's a stop which checks for damaged DNA. The S phase, there's another stop that checks for unreplicated or damaged DNA. Same thing kind of goes for the end of the G2. And then finally, the M phase is one where it's looking for chromosome misalignment as well as attachment. So this is basically a general rundown of the cell cycle. I hope you all found it useful, and as always, best of luck studying.